Okay, so I'd like to start off by thanking Andy for inviting me here. It's quite a bit outside of the scope of the usual meetings I go to, but still, I hope I can contribute to the antibody validation topic by telling you a little bit about our efforts to look at post-translational modifications, and in particular on a protein that's not a histone. So we've actually, yesterday in the panel, one question came up. Well, is there even a role for antibodies in the PTM toolbox? Can't we do everything by MS? And while MS certainly has a big role in looking at these modifications, I think that antibodies are still very important both for the identification of individual PTMs, but also looking at different coexisting combinatorial PTMs, which is often hard to do when you do mass spec and you actually fragment your protein. But also for doing specific functional assays that actually relate to what the PTMs do to the protein to change its function or localization. So we, of course, use a lot of different quantitative and analytical approaches that use antibodies, from Western blots to ELISA microscopy, flow, IP, and probably other things you can think of. And what my team is interested in is to look at the regulation of protein function by PTMs. So there are very many beyond phosphorylation that can determine which binding partners a certain protein will have, and depending on the binding partners, it might then lead to very different biological outcomes. And of course, the one very famous example is the histone code, where you can have very different modifications on mostly lysines of the histone tails that will lead to either activation of gene expression, repression of gene expression, depending on the readers that will interpret this code. But it's not only histones. One other protein that's famous for PTMs is p53, which has phosphorylation, acetylation, many different modifications that will determine its function. And my team is working on the microtubule binding protein tau, which is another one of these heavily modified proteins. So as you can appreciate from this cartoon, tau is subject to very many phosphorylation events. We have a lot of acetylation that has been reported. Methylation has been seen where nobody really knows what it does. We have ubiquitination, which is sometimes counteracted by sumoylation. We have a sugar modification that has been reported. So it's a big, complicated mess, really. And the question is there a tau PTM code? Is there something analogous to a histone code that will determine tau's localization, tau's function, that is mediated by all these different modifications? And why is tau even important? So tau dysfunction is a hallmark of many neurodegenerative diseases, and one of them is Alzheimer's disease, where tau aggregates are one of the hallmarks of the disease. So you might be familiar with the extracellular amyloid plaques, but there's also intracellular protein aggregates that actually consist of hyperphosphorylated tau protein, and that spread throughout the brain during the disease. And the brain regions that are affected by this tau phosphorylation, they correlate very well with the phenotypes of the disease, so the spreading correlates with the memory decline in the patients. But also other PTMs are strongly linked to Alzheimer's disease, and it's slowly coming up in the literature that it's not only all about phosphorylation. So we have papers about tau acetylation that can promote aggregation. We have sumoylation, as I said, that shows an interplay with ubiquitination and can regulate tau degradation. We have proteolysis, which is another big factor in tau pathology, and glycosylation, which seems to be somewhat protective against aggregation. So our vision is really to identify a PTM signature for tau, and to see if we can find one that correlates with Alzheimer's disease states, because then you could exploit the signature and look for potential therapeutic targets. You might also be able to use it as diagnostic markers and so on. So we started out this project about 2.5 years ago by looking at, well, what PTMs are even reported on human tau. And we assembled all of this information into a database, also to visualize it, where they are on the protein. And we came up with a total of 92 different modifications that were found from human brain tissue. So there's more that are reported in other model systems, but we wanted to focus on the human. And then, as I mentioned, there are mainly two ways to analyze these things. There are antibody-based methods and there are non-antibody-based methods. And because this is an antibody meeting, I'm going to focus on our work with antibodies. And we found that about 1/3 of all the modifications were covered by commercially available antibodies. So while this is not a bad number, it is mostly phosphorylation sites, because phosphorylation is the most well-studied PTM on tau. So there is a bias in this work towards phosphorylation, but yes, we're trying to cover the other modifications by mass spec approaches. Oh, yes. So, and we stumbled into this a little bit blindly, I have to admit, by just buying a bunch of antibodies that are out there from different vendors towards tau phosphorylation. And the first question that we asked ourselves is, well, can we actually trust these? Do they really only detect the phosphorylated tau? Or is there a danger that we might confound our data by also looking at non-phosphorylated versions of the protein? So, and especially talking to my friends from epigenetics, I realized, oh, in the histone field, they have these peptide arrays where they really judge specificity of antibodies based on binding to modified or non-modified peptides. So we decided to design a peptide array based on the tau sequence, and we introduced modified amino acids in this array. So we walked through the sequence around the modification and modified the amino acid in question. So this is a nitrated tyrosine here. And sometimes we also introduce off-target modifications. So here we nitrated another tyrosine that's not supposed to be detected by the antibody. And this is actually an example of a very good result, because the antibody specifically detects always the peptide that has the tyrosine modification in question and not the other one. But then, of course, there are also other examples. So this is an example of a phospho site where we tested 5 different antibodies. They're all from different vendors, and we assume they are different antibodies, although you can never be sure. And again, we have this phosphosite, phosphoserine here, the red one that's supposed to be recognized, and there are other serines and threonines in the surrounding that could be phosphorylated. So while these antibodies seem to detect the peptides they should, there are some very strong cross-reactive dots here, and especially here the non-phosphorylated peptide, but also sometimes the neighboring threonine when it's phosphorylated are detected. Or here another one on spot 6. This is also a non-phosphorylated peptide. So we stumbled across these very worrying results. They were very worrying to us, and especially for this phosphosite, I have to say we did not find a single antibody that was actually specific enough for our applications. So this makes you, of course, also doubt data generated with these tools and so on and so forth. But we decided to kick out this phosphosite from our repertoire because we just didn't have a good tool for it. And then there are also complicated cases. So as I mentioned, tau is very heavily modified. So it could happen that you have 2 amino acids that are modified that are sitting right next to each other. How would antibodies deal with that? Are they hindered by the additional phospho group? and some of them are. So here you can see this antibody very nicely recognizes many of the peptides that are modified at the correct site. But then it doesn't recognize the very same peptide when you introduce a tyrosine phosphorylation just adjacent to it. So these are also things you need to keep in mind when you're using these tools, that if you have a heavily modified protein, detection could be hampered by additional modification. So the answer to how specific are these antibodies is really, it depends. And keeping all this in mind, we assembled a big long list of all the antibodies that we tested, and this is published. But in general, 50% of the ones that we tested turned out to be non-specific. So we thought, well, this is actually a significant problem, and that was also one of the reasons why we decided to publish this, because I don't think, or at least talking to my colleagues, I don't think that this problem is very much appreciated in the community. We then went on to test whether the antibodies are actually specific for tau, because of course a peptide is the one thing, but you wanted to recognize the whole protein. So we got tissues from transgenic mice that express human tau, and we had the corresponding knockout mice that are knocked out for the mouse tau. And luckily, all of the antibodies that passed our peptide array criteria also passed the Western blot criteria with these tissues. So this is just a general overview. Sometimes we saw very faint bands in the knockout, but the majority of the signal was really specific to the transgenic mouse brains. And because we wanted to use these antibodies, so this validation was really a means to an end, we wanted to get to the point where we could analyze actual patient tissues and get on with the project. So we decided to set up an ELISA using these antibodies to increase our throughput a little bit and also get some more quantitative data. And so we used our antibody panel and coupled the antibodies to streptavidin plates. Then we would run our analyte, which was mostly human brain tissue in this part of the project, and then use a total tau antibody for detection. And doing this, we found, again, with human transgenic tau mouse brain and tau knockout mouse brain for controls, we found that most of these antibodies had very low background signal in the knockout, and we got a decent signal from the transgenic mice. And a similar picture emerged for the phospho-tau antibodies, although many of them have a variable baseline, so there is some background signal here, but we still think the assay window is large enough to allow for detection of the modified species. And if we're comparing between a disease state and a control state, we would never expect the control state to be completely devoid of PTMs anyway, so we're looking at a difference in signal, really. So we concluded that, okay, our antibody panel is now ready for showtime. We want to analyze some real tissues. And we decided to go for different brain regions and also different disease stages. So the way tau pathology progresses throughout the brain and the disease is grouped into the so-called Braak stages. So you have a prodromal phase where the patient actually doesn't suffer any memory loss, and the tau tangles are really confined to a small area of the brain, the entorhinal cortex. Then in the early disease stage, which is Braak stages 3 to 4, you have spreading to the hippocampus. And then in the late stage disease, you really have most of the cortex affected by the disease. So we took brain samples from aged people, so these are age-matched, at Braak stage 0 to 1, and then at Braak stage 3 to 4, because we wanted to look at early disease rather than a very late stage where neurons would already be dead and lost. And this is the kind of data that we generated. So for the phospho panel, we got signal for all of the antibodies. And sometimes you could really see a nice separation between the BRCA3 and 4 compared to the BRCA 0 to 1. And we did this exercise with 3 different brain regions, and from the same patient, though. So from the same patient, we took the strongly affected entorhinal cortex, we took the intermediate hippocampus, and then the temporal cortex, which we would not expect to be strongly affected by tau pathology at this disease stage. And we generated heat maps, and you can immediately appreciate that some antibodies actually give us a very strong signal. And I'm always, in Alzheimer's meetings, people always ask, oh, that means there's a lot of the PTM there. And I always have to say, well, it also depends on the affinity of the antibody. So we cannot really tell which one it is, other than we get a strong signal. And what was really striking to us is that, especially in the hippocampus, you get a very strong separation in many PTMs between Alzheimer's and controls. And in the temporal cortex, the pattern sometimes continues, so you sometimes still see differences between Alzheimer's and controls, but it's not as strong as in the hippocampus. And when we look at this data in a slightly different way, and we can look at the significance of the difference between the different BRCA stages and also the fold change in the volcano plot. And you can see that we got some samples from BRCA2 staged brains, where you can see that actually none of these values cross our significance threshold. So this was set at 0.05 as the threshold, which is the red line here. But once you start comparing BRCA 01 to BRCA 3/4, you can see that 4 of these PTMs move across the threshold and are more enriched in the Alzheimer's brain samples. So there's really a transition between these BRCA stages, also in terms of tau PTM pattern. And when you now look at BRCA 01 versus BRCA 3/4 in the different brain regions, you can see that the hippocampus, as already suspected by the heat map, shows a lot of changes, whereas we got just 4 of them in the entorhinal and 2 of them in the temporal. But what was also interesting to us is there are commonalities. So these 2 actually show up in all of them. And these 4 are also a subset of this. So it's very consistent, and there are certainly some changes that are always associated with the disease state. So, of course, the next question now is, are these disease-associated PTMs, are they drivers or are they symptoms of the disease? And how can we analyze this? Well, it's not easy to do with the tissues that we've been working with so far. So we really need to move to a cellular model. And to do this and to culture human neurons in a dish, the state of the art is to use iPS-derived neurons. And we have set up iPS-derived neuron cultures and set out to do experiments to screen for modulators, to look at the impact of stressors, to look at the effect of PTMs on tau, localization and function. But with our experience coming from the antibody validation in the first part of the project, we asked ourselves, well, are the antibodies still specific also in the iPS model? Because we hadn't tested that yet. And actually, we were super excited by some early immunofluorescence work using our antibodies, because we could see some very different staining patterns. So if you look at nitrated tau, you can see staining of both neurites and cell bodies. If you look at acetylation, you can see mostly the nucleus is stained, whereas the tyrosine phosphorylation here, you can see neurite staining. But How specific is this? So we decided, okay, we are dedicated to this validation business now. We need to make a knockout. And we generated CRISPR knockout in our iPS cells by using 2 guide RNAs that would cut out a part of exon 1. And so by cutting out this part of exon 1, we can screen our clones and we can figure out who's a knockout by running PCR products of exon 1 on a gel. And if the band is small, it would be a knockout. If the band is large, it would be a wild type. Sometimes you get both, which means it's a heterozygous clone. So we really think that on the genetic level, we have knocked out tau. And we thought, okay, great. We will just run some Western blots with total tau antibodies to verify. Yes, so these antibodies have their epitopes spread out all over the protein. And most of the time, you get a strong band in the wild type and little to nothing in the knockout. But there are some exceptions. And especially, we were worried about this one band that kept popping up just a little below the wild type. Because we thought, oh no, we're cutting out exon 1. Is there an alternative start codon maybe that will make a truncated version of our protein? And because it was picked up by 4 out of the 6 antibodies that we tested, we cut out the band, sent it to mass spec. There was absolutely no tau in there. So the conclusion that I have for now is that based on the genetic data, based on the mass spec data, our knockout is a knockout, but still 4 out of 6 antibodies give us a band. So yeah, so far I think it is a knockout, but especially because we have an antibody that's directed against the C terminus, which if it was an alternative start codon should still be there. So we think that this band is really a cross-reactive, non-specific band. And looking at immunofluorescence, actually the picture is very similar with these non-specific antibodies because you get, in the red staining here, your tau antibody. It looks very much the same between wild type and knockout. So moving forward with our experiments, we really, based on this results from this knockout cell line, we try to choose carefully also our total tau antibodies, because when we go into screening methods, image-based screening or flow-based screening, we need good negative controls, and we think we have generated them. So in summary, I really have to say that we've encountered significant issues with many commercial antibodies, but that being said, I also have to say that for the big companies, customer support has been very helpful, and there's been a lot of back and forth. 50% of actually the PTM tau antibodies that we tested were not useful for us, but also different applications require different tests. I mean, this has really been a consensus throughout the meeting. And the validation methods that have been very helpful for us were mass spec and also knockout or knockdown. And the big lesson for me was that cells give very different results from tissues. So with that, I would like to thank the team and our collaborators. Actually, the brains came here from the UK, from Brains for Dementia Research. And yes, Ebru was the driving force behind all the validation. David is the master of IPS, and Yasmin is helping him with the knockout. So yeah, thank you.
I just found really interesting your data on when you have multiple modifications quite close. Because actually that happens also in histone modifications.
Yes.
And one thing we have found is that depending on how you immobilize the peptide to the array, it could have quite a lot of difference. So it could be the assay
Yes.
as well as the antibody, so to speak. But so we, in general, try to do a couple of different assays or immobilize the peptide with linkers.
Right.
Or,
Yeah. Yeah, one thing I've noticed is also that it depends where in the peptide, of course, the modification sits. So we try to walk it throughout the peptide because sometimes it's closer to the membrane, sometimes further away, and all these issues. But yeah.